Hello, I'm Kyle Havel,
I'm Katie Perotti.
And I'm Alexa Nemeth.
and this is our meningitis vaccine proposal. Meningitis causing pathogens exploit host microbial interactions and signaling pathways to breach the blood-brain barrier. Vaccines have substantially decreased bacterial meningitis rates in the Western countries over the last 2 decades, but impoverished countries still deal with the disease. Meningococcal disease caused by N. meningitis and Streptococcus pneumonia manifests usually in children and young adults with symptoms such as migraine fever and stiff neck. Vaccination initiatives recommend MB and MACWY vaccines. Protective immunity is achieved through antibody productions facilitated by vaccines. Vaccine efficacy assessment involves preclinical animal testing and FDA-approved clinical trials with 3 phases. A randomized controlled trial conducted in a blind manner with 2-dose administration and a follow-up blood test is crucial for determining vaccine efficacy. Analyzing human serum bactericidal assay HSBA, factor H responses, and measuring meningococcal antibodies to determine absolute vaccine effectiveness. For our literature review, meningitis is characterized by inflammation or infection of the cerebral spinal fluid surrounding the brain and spinal cord. It is a complex condition orchestrated by pathogens employing intricate strategies. These involve host and microbial elements, navigating signal transduction pathways to breach the formidable blood-brain barrier. Meningococcal vaccines have notably reduced incidence, especially in high and middle-income countries, but a dichotomy persists with Western nations experiencing substantial declines, with African countries facing higher risk. Key survival strategies involve factor H binding proteins, FHBP, and type 4 pili, TFP, where FHBPs bind to factor H enables immune evasion, and TFP's filaments facilitate initial host interactions. Understanding these molecular intricacies is vital for targeted interventions. Notably, vaccines and ongoing global collaboration is imperative for addressing this enduring health threat. Our figure 1 is basically how meningitis enters the body. It enters through a mucosal body, such as the nose or the intestines, anywhere where mucus is present. And it does that by evading, entering the bloodstream, and then eventually binding and entering the blood-brain barrier.
So for vaccine design, in order to create a protein vaccine that targets meningitis B, we plan to isolate the recombinant proteins found on the surface of the pathogen. And the proteins we plan on isolating are FHBP, or factor H binding protein. It's a surface exposed lipoprotein that binds to human factor H. NAD-A, or nadiryl adhesion, which is another surface protein that mediates pathogen entrance to the nasopharyngeal epithelial cells, and OMVs, or outer membrane vesicle proteins that are released by the pathogen. And once we have those proteins isolated, we would add an aluminum adjuvant, which would help make the vaccine last longer and make it more effective. And this vaccine would be targeted towards children and young adults, and injection for this vaccine will occur intermuscularly in the deltoid muscle of the upper arm. And for figure 2, you can kind of see that FHBP and NAD-A are all surface proteins on the outer membrane vesicle that comes from the pathogen.
Now, for the efficacy testing of our vaccine, the first thing we had to do was a safety trial, which includes an animal vector. In this case, we used a mouse, which must be used to determine the safety of the vaccine and making sure it follows FDA regulations for human use. And then once it passed, we went on to phase 1, where there's a clinical trial with about 20 to 100 people of good health and around the same age. And in this phase, we were able to identify side effects and study the subsequent immune response from the vaccine. And for phase 2, we would vaccinate a few hundred healthy teens to young adults, which allows further examination of the side effects. and reactions to the vaccine. Once phase 1 and phase 2 are passed and the vaccine seems okay, we will reach phase 3, where we will vaccinate thousands of participants, which this is a confirmation phase where the vaccine's overall effectiveness and efficacy support the claim to safety. If the vaccine is proven effective and safe, the FDA will approve the vaccine. In our figure 3, this is a experimental design of the mouse immunogenicity models. This is why we chose mice, is because humans are the only natural host for N. meningitis, but small animal models such as mice have been developed. Mice have been chosen in this trial because they have shared biomarkers, in this case, IgG responses, so antibody responses to humans in relation to meningitis immunology. Using mice, as shown above, allows for examination of an optimized immunogenicity model, which denotes the dose regimen for optimal immune response.
And while vaccines are important, they are unable to prevent all cases of meningococcal disease. So vaccination and symptom monitoring is recommended for all individuals, especially those living in high-risk areas. And the development and implantation of this protein-based vaccine, along with the continued research and surveillance, are crucial steps in the ongoing effort to prevent and control meningitis. Thank you.